My name is Jairo Arroyave. As Liz mentioned, I'm at Instituto de Biología, UNAM, in Mexico, Mexico City. And today I'll be talking about phylogeography of fishes from cenotes and flooded caves of the Yucatan Peninsula in Mexico. Let's see. So the Yucatan Peninsula in Mexico is a karstic region. And for those of you not familiar with the term karst, it refers to topography dominated by limestone and other soluble rocks that is characterized by the presence of different kinds of structures that result from that dissolution process, the dissolution of the rock. So you can find caves that can be either dry or flooded, sinkholes, fissures, and other features of this landscape. And normally you find karstic aquifers associated to these formations. And this groundwater has been colonized by various groups of organisms, including fishes. In Mexico, roughly 20% of the continental territory is dominated by this kind of landscape, of which perhaps the Yucatan Peninsula is the most famous, possibly because of the presence of cenotes. And cenotes and submerged caves are pretty much the main fresh waters that you find in this region. This region lacks pretty much pretty much throughout the entire region, especially the northern half. This region lacks rivers and streams or lakes and all forms of surface water. So what we have is basically these formations that are cenotes, which are the part that we see here, which is the collapse of the rock after the dissolution that exposes the water in the aquifer to the surface. So these karstic windows are what we call cenotes. And in this map, you can see that most of the cenotes, these tiny dots, black dots on the northern half of the peninsula, are the main bodies that we find here. We can find probably thousands of cenotes throughout the region. And as I was saying, they basically connect the water of the aquifer to the surface. So you can see these different habitats: the cenote pool, the cavern, and the cave. Caves, really the way they have been documented for the most part is through the expeditions and maps and mapping activities by cave divers. And because it's so technical and so really hard to do this work, there's not a lot that we know really yet of all the area of the Yucatan Peninsula when it comes to the underground architecture of the aquifer. However, cave divers have documented a bunch, especially in the Riviera Maya region. And actually the longest underground, underwater, the longest flooded cave is located in this region. It's known as the Sistema Zacactun in Quintana Roo. Cenotes come in different shapes, different forms, depending on the way they form. And here are some nice pictures of cenotes. You can see the cenotes of the open type, where you can see that the limestone rock that has collapsed in this section. Some other cenotes look like lakes, like this one here, with a lot of vegetation associated to them. These are some examples of flooded caves, cenote cavern type. Another one where you can reach the water by walking through a dry cave area. And well, the peninsula emerged from the ocean around 3 million years ago or so during the Pliocene. However, the system I'll be talking about today is much younger. These caves that are now submerged in the cenotes were really dry until like 20,000 years, when the maximum, like the last glacial maximum, when we started to see a rise in sea levels, and all these dry caves eventually flooded and formed the system that we have today. The ichthyofauna that inhabits this region is not necessarily super diverse, but is like moderately diverse. We have 22 species. It's interesting from other reasons. Beyond diversity, we have like a good phylogenetic component, different orders, 6 orders, each with one family, and 4 endemic species, 2 of which are cave dwelling. So we have here our troglomorphic species, the blind swamp eel of Easterna infernale, and the Mexican blind brochure, Typhias piercii. Most of the species of fishes that inhabit this system, however, are really more associated to the cenote pool. They tend not to venture much into the cavern or cave, at least that we know of. And these are examples of some of these species, including the catfish Grandia guatemalensis that I'll be talking about today. And so that strong affinity to the cenotes, to the cenote pool versus the submerged caves that most of these fishes exhibit poses a very interesting question from an ecological and evolutionary perspective, and that is how genetically structured are these fish populations from different cenotes across the Yucatan Peninsula. Now, you would assume that gene flow between fish populations from different cenotes that on the surface seem kind of isolated would be contingent upon the degree of underground physical connectivity and the capacity of the species themselves to move and disperse through these conduits. So if we have those two assumptions, the connectivity assumption and the dispersal capacity assumption, we could frame two working hypotheses. One would be a hypothesis of panmixia, basically that we have gene flow across all these populations of the different species that inhabit the region, despite the seemingly localized and isolated nature of these habitats. And the other hypothesis that we could come up with is that of phylogeographic concordance, to see if co-distributed fish species would exhibit concordant phylogeographic patterns as a result of similar responses to the same historical determinants. There's really not much research that has been done in that system, at least prior to when I became interested in this research. These are pretty much the two only papers that I could find that somehow address these questions. The last one is much more recent. It's from 2 years ago. It's about this Mexican cichlid Jairo Arroyave in the Yucatan, not only in the cenotes, but also other freshwater bodies in the peninsula, especially in the southern portion, which is less devoid of surface water. And so we decided to address this question and shed some light into this issue that has not really been explored much previously, with research first on Grandia guatemalensis, a catfish species that is widely distributed not only in the peninsula but throughout most of Central America and part of South America. So this catfish, as I was saying, has a widespread distribution that goes from the south of Mexico to way to northern South America. Here are some pictures of the fish, so you can see how it looks. When it comes to phylogeographic research on these species, really the only work or like the main work that you can think of is one paper published in 2002 about the taxonomy of Central American Rambia that included populations of Rambia guatemalensis throughout most of its geographic range. However, it didn't include populations from the peninsula. So with this, partly one of the reasons we chose this fish to investigate genetic structure in the peninsula was the ubiquitous nature of the species throughout the cenotes, but also that it would be interesting to see what happens with this section in the context of the entire radiation across the species range, because that hadn't been explored before. As I mentioned before, this species is very common throughout the peninsula in lots of different types of cenotes. It's one of the easier fishes to find in the region. And so the study that we conducted consisted of a sample of 47 cenotes, are the red dots here that you see in this region, a few here in the middle, and others in the Riviera Maya region. For a total of 90 individuals, we sequenced mitochondrial genes, 3 in total, and we perform a series of phylogeographic and population genetics analysis. Now, we were not going to compare. It was really analytically challenging to compare all 47 cenotes and assume that each was a separate population. So that implementation was really not feasible. So we decided to have an approach in which we would use regional scale fracture zones as like to assign these samples. in surface waters, normally you use drainage basins as units of comparison, but in this case we don't really have a lot of that information. So the closest that we could come up with for drainage basins were these regional scale fracture zones that had been in the past proposed by geologists. So that gave us like a broad idea of patterns of subterranean water flow, and hopefully we would find some kind of genetic structuring that was related to that in case there happened to be such a structure. So these are basically the main results from that study, in which we did find a striking genetic correspondence, genetic signal and structuring of these a priori designated physiographic regions in the peninsula with these major haplogroups. This is like the haplotype network resulting from those data, and the gene tree resulting from those data. And we see that for the most part, there is a strong phylogeographic region that for the most part, as I was saying, coincides with those regions. We complemented this analysis, this gene tree and haplotype network analysis, with population genetic analysis, and those also supported this idea of significant genetic structure between these physiographic groupings using either FST or AMOVA analysis. Now, in the context of the entire radiation of the species, which the Yucatan Peninsula was absent before in those analyses, we find interesting things here. Before we run this analysis, I thought that maybe the peninsula was going to be somewhat isolated and would have its own history independent of the other regions. And we ended up finding that a number of different basins, including some inside the peninsula in the south and others outside of the peninsula, actually had samples that resulted very closely related to those with the aquifer. So like well nested within our aquifer samples. And that the pattern that we recover from these, which I'm not showing here the details, but this is just the summary trees, but could be consistent at least with a history of colonization and of one single colonization event and multiple dispersal events out of the peninsula. Now, in addition to Rhamdia, we started working with Rhamdia, with Tiflias, with the Mexican blind brochure, very photogenic, as you can see in these beautiful pictures from a couple of cave divers that have been assisting me in this work. Basically, the idea was to conduct a very similar study. We have this species that is widely distributed in the peninsula, is endemic of the peninsula, is not found anywhere else. Most likely, this is derived from a marine ancestor because this group is primarily marine. These brochures, the work that had been done in the past really didn't include any phylogeographic kind of a study. It was mostly basically the placement of these species in the context of the phylogeny of all viviparous brochures. And so we had a sample also very thorough across the peninsula. In this case, we had less cenotes, 32. Sampling these individuals is much harder, as you'll see. We had almost 90 individuals, 3 genes again. In this case, we used 1 nuclear, although it didn't show a lot of variation. And so far we have some gene genealogies. We're in the process of the other analysis. This is the sampling that I was going to tell you that is not trivial, and it requires like highly specialized and technical cave diving skills. This is also to show you how the habitat of this fish looks like. This cave diver is probably around 25 meters deep and 40 meters into the cave. And this is how we do it. And the results so far show a genetic structure that is not as clear, at least using these units of comparisons, as with Rhamdia. But we do see some structure in that we're still further exploring with the analyses that are yet to come. So to conclude, we can say that a striking spatial correspondence between major mtDNA lineages, at least in Rambia guatemalensis, and physiographic regions within the northern half of the peninsula, um, based on broad scale patterns of groundwater flow. So there's something that had not been documented before, and at least for Rambia guatemalensis is very strong. For the other species might not be as strong, but we're in the process of testing really that with more analysis and data. Regional scale hydrogeological differences might impose limits to underground dispersal, potentially driving genetic divergence in fish populations inhabiting this aquifer. Our findings add to a growing body of evidence in support of the notion that the architecture of karst aquifers may include physical barriers that promote genetic structuring of the stigofauna. And lastly, as a relatively nascent field of research, we hope that these studies will pave the way for future research devoted to unraveling the evolutionary history of the Yucatan Peninsula stigofauna, not only ichthyofauna, but there's other groups of other taxa that inhabit this system, a number of crustaceans and other invertebrates that are also troglomorphic. And with this, I guess I finished my talk. Thank you so much for your attention. And let's see if we have some time for questions. I'm not sure.
Great, thank you so much, Jairo. That was awesome, and I love to see the videos as well. Yeah, we have about 4 minutes for questions, so feel free to put them in the chat if you got them.
Should I stop sharing my screen?
Sure. Yeah.
Can everybody see me now?
Yeah. Okay. So.
Perfect.
Jairo, you see that first question from Moises?
Yeah, from Moises.
Yeah.
Jairo, how active are these individuals? Are they easy to collect with just a hand net? Well, Rambia, the catfish we primarily rely on these traps, these minnow traps, but we also sometimes use other kind of nets, including cast nets when we can do the cast netting. But yeah, they're very active, but they're relatively easy to catch, especially with the traps. Now and as for the Tiflias you saw in the video, like as long as you get to the habitat, it's really not that complicated because they're not terribly skittish. But the challenge is to get into the caves. That's the more challenging part. Now, Darko, given the high genetic structure in the catfish, even within the Yucatan Peninsula, why is it still considered a single species? Well, that's an interesting question. That one thing, you can have a very high genetic structuring and still have the same species. We don't necessarily have evidence, at least in this study, that is suggestive of underrepresented diversity of species. We still consider this is the same species, just like this species is highly movable, has like a very widespread distribution throughout the continent. So I think in this case, it's just the fact that there's not enough morphological differentiation. We also did morphological work, not included in this presentation, but and actually when it comes to morphology, there's not a real signal of differentiation. So the signal was mostly within the mitochondrial DNA, and that's normal for species. There's always genetic structure, not always, but some have a higher degree than others, so that is not necessarily evidence for taxonomic differentiation. And the other, I've heard that the ring of cenotes is defined. Yeah, that's true. That is yeah, I don't know the details of how that impact crater somehow influenced the presence of these cenotes. I don't know if it has to do with making that terrain more like somehow easier for it to dissolve in the way that these formations develop, but yeah, I don't know much beyond that in geological terms. So I guess that's it.
Awesome. Thank you so much. So it looks like
wait,
we have one
I
more
think there's
question
one
from Chris. Yeah.
Do we have time? Yeah.
Yeah.
Hi, Chris. Great talk. Do you think the population structure lack of speciation will compare for the more troglomorphic? Well, that's a very good question, and I actually think that the pattern that we're seeing so far, we still have to do more analysis with the brochure. seems more homogeneous than the one we have for Rambia. And I think that makes sense in the if you think of the dispersal capabilities that the brochulas would have, given that they are highly adapted to these hypogean habitats. So like from that perspective, you would think that they would be more capable of moving across these underground conduits and these submerged caves, and therefore contribute more to gene flow and homogenization of the genetic variation. So I guess that's it. Yeah. Thanks, Liz. I think that's it.
All right. Awesome. Thanks so much. Thanks, Jairo. Okay, so next we have Pam Hart from LSU, who will be talking to us about other caves in North America. Take it away, Pam.
All right, thank you. Okay, hopefully everybody can see this. Thank you so much for the introduction. Thank you all for being here, for Zooming in today. I'm excited to share this project in which I looked at the evolutionary history of cave-adapted Amblyopsid fishes. So I think I'm in good company saying that weird animals in extreme environments are pretty cool. We're drawn to study these organisms in places like deep, turbid, fast-flowing river channels, the deep sea, islands, deserts, caves. These are all geographically isolating locations that impose harsh physiological demands on resident organisms. And within these environments, we find examples of convergent evolution. Convergent evolution, or the independent evolution of similar traits in species of different lineages, can be seen here in deep sea animals with modified vision, marine mammals with streamlined body plans and the loss of legs, and cave animals with the loss of eyes and pigment. Now, these last two are examples of convergent evolution with the loss of a complex trait. And this is actually a lasting topic in evolutionary biology, whether or not the loss of that complex trait is reversible or not. In 1893, Dolo said, no, once that trait is gone, it cannot then be regained. I was in particular drawn to research, as many of you were, by caves and cave obligate organisms, because I could study those topics that I've mentioned. We have animal adaptations to harsh environments. I think we can all agree that caves are pretty gnarly. We have the loss of a complex trait, including eyes. Eyes are pretty complex. And then the reversibility or irreversibility of that trait loss. And in particular, I use fishes from the North American family Amblyopsidae to study these topics. This family includes surface-dwelling species with well-developed functional eyes and vision, and cave-dwelling members with no external eye characters and lacking visual perception. They are distributed throughout the southeastern United States. And I would like to point out here that I have colored the text related to eyed fishes in orange and blind in blue. And this color coordination for eyed and blind will continue throughout the rest of my talk. So it's a really great system, but of course things aren't that simple. It appears that the pathway to cave adaptation has not been straightforward in the family. Here we have the top two phylogenies created using morphology, in which we could see what's considered a simpler path to cave adaptation, in which we have the eyed forms as united and the blind fishes as monophyletic. And with these phylogenies, we come to our first ancestral eye state hypothesis. So how did they lose their eyes? Well, in this particular case, it is suggested that we have a progressive loss of eyes. So a stepwise loss from larger eyed fishes to small eyed fishes to no eyed fishes. And this stepwise loss is known as progressive regressionism. There is, however, disagreement between morphology and molecular. We do have phylogenetic discordance. These two phylogenies here on the bottom show what could be considered a more complex pathway to cave adaptation, where we have the possibility for multiple eye loss events or even surface recolonization and eye redevelopment after eye loss. So taking a closer look at one of the more complex options, this is the multi-locus species tree from Niemiller et al. 2013. And what was interesting, firstly, was that they recovered an eyed form within the group of larger eyeless or blind fishes. And using ancestral state reconstruction, they found support for a blind ancestor here, followed by eye redevelopment in this eyed form. So firstly, that's interesting because it would be the reversal of complex trait loss, therefore breaking Dollo's law. Dollo's law has been challenged before by things like oviparity in snakes and eyes in ostracods. But it was additionally interesting in this paper because further data, including opsin gene mutational data and eye histology, both suggested multiple eye loss events rather than the eye redevelopment. And so this brings us to the 3 ancestral state hypotheses for this family, how they lost their eyes. Firstly, the stepwise loss of eyes. Secondly, an individual cave colonization event followed by surface recolonization and eye redevelopment, or multiple independent eye loss events in cave invasions. So the Amblyopsids are interesting for their evolutionary history, but they are also interesting for their morphology, and not just because some of them are eyeless, but rather they show this body shape convergence that's seemingly common among aquatic cave obligate vertebrates, as seen here on the left in an Amblyopsid cavefish and on the right in a plethodonid cave salamander. You may notice this flattening of the head, and then they have duckbill-like snouts. This shape variation has been noted, but has rarely been quantified. And so being interested in convergence, I wanted to also look at morphological convergence in non-sister cavefishes using shape variation. And so this brings us to the objectives of this project. It was to firstly use genomic data to clarify the evolutionary relationships in the family. Secondly, to determine the most likely scenario for ancestral eye state. And lastly, to evaluate the effects of cave adaptive evolution on body shape variation. For the phylogenomics and ancestral state reconstruction portion of the project, me and my co-authors sampled all Amblyopsid genera and used the closest relatives as outgroups. For shape variation, I chose to use geometric morphometrics, and I had 2 data sets. Firstly, one of 135 total museum specimens, and then I had a voucher data set in which I had identical matches between the morphological and molecular data sets. So I was able to incorporate phylogenetic relationships into the shape data. For the sake of time, I'm not going to get too much into the methods. Additionally, I've had to decrease the amount of results that I have presented here today, but this paper is out in Evolution if you would like to take a look at it. So I began with the target capture of ultra-conserved element loci, and this is a popular phylogenomics technique. I then used multi-species coalescent analysis to create phylogenetic trees, and then I created a phylogenetic network using splits network analysis. We performed ancestral state reconstruction using a Bayesian framework. I then tested for differences in shape between eyed and blind fishes in the full data set using a MANOVA. And I incorporated phylogenetic signal with the voucher data set using a phylogenetic MANOVA. Lastly, I examined morphological convergence using Staten's convergence index in the non-sister cavefish lineages. These are the multi-species coalescent trees, both created with a 75% complete data matrix, which equates to 897 UC loci, and they both do agree in topology. And what was particularly exciting for me was finding the eyed spring cavefish as nested within the group of blind cavefishes. And so our phylogenomic topology agrees with that of Niemiller et al. 2013. This is the splits phylogenetic network. Each split indicates a non-tree-like or non-bifurcating signal. And so since we have splits, we have non-tree-like signal. Some of that signal is in a lot of the cavefishes, but also we have some interesting non-tree-like signal between blind and eyed fishes in both cases. So these relationships could be caused by introgression, incomplete lineage sorting, or hybridization. With this particular splits network, I can't support one of those scenarios over another, but hopefully maybe stay tuned to my research. I am looking further into this. For our ancestral state reconstruction, we tested 3 different models. Firstly, that all transition rates were the same. So we had the same between blind to eyed and eyed to blind. Our second model was that all transition rates were different. And then our third model, we specifically tested for irreversibility. So we tested for Dolow's law. And this was the chance that a transition from blind to eyed was 0. And the model that we found the most support for was that of irreversibility. So taking a closer look at this, we found support for eyed ancestors at these 3 nodes, indicating at least three eye loss events, and thus convergence on a blind state in the non-sister cavefish lineages. With our posterior probability, we found about 66.7% of our 10,000 runs, we found no transition from blind to eyed. On to shape variation. This is a principal components biplot, PC1 on the X, PC2 on the Y, and this equates to about 58% of all shape variation. Blind fishes tended to have longer heads and narrower bodies, and eyed fishes tended to have shorter heads and deeper bodies. I did find significant differences in shape between blind and eyed fishes using all of shape space. This is the GM phylomorphospace. So it is a PC biplot, just like the last one, PC1 on the X, PC2 on the Y, but it has the phylogenetic relationships mapped onto it. So I did find that phylogenetic signal had significant impact on the body shape of these fishes. But after accounting for that phylogenetic signal, I did still find significant differences in shape between blind and eyed fishes. Lastly, I did find support for shape convergence among the cave obligate fishes. I found an average of 17% convergence, which represents about 10% of all morphological evolution in these lineages. So to conclude, we have well-supported phylogenies in which we recovered the eyed spring cavefish as nested within the larger group of blind cavefishes. Using splits network analysis, I did find evidence of non-tree-like relationships and signal, but at this point, I can't determine which biological event caused those relationships. In our ancestral state reconstruction, our most highly supported model was that of irreversibility, and we found evidence for 3 independent cave colonization and eye loss events. Lastly, I did find significant differences in body shape between eyed and blind fishes in both the full data set and the voucher data set. And I did find evidence for convergence in the cave obligate fishes. As for significance, with this study, we've added additional support for multiple eye loss events in cave obligate taxa, rather than support for progressive regressionism or eye redevelopment. This has been seen previously in other cavefishes, as I'm sure we'll hear about later, as well as cave-adapted brittle sea stars, which I thought was pretty cool. I did find that cave adaptation has an effect on the body shape of fishes, and that cave obligate fishes are converging on similar shape. Lastly, I'd like to point out how important it is to use multiple lines of evidence when looking at cave obligate taxa because of the complex nature of cave adaptive evolution. I want to thank all of my co-authors, the museums especially for all of their museum specimen loans, and my funding resources. Please feel free to contact me if you have any questions afterwards. And thank you all so much for your attention.
Great, thank you so much, Pam. That was amazing. So once again, feel free to put questions in the chat and we'll go through them as they come up. So you have a question from Darko, do you see it?
I do. Okay, do you know how long ago these caves were colonized? Did it take the same amount of time on each case to lose eyes on each species? So the family started diversifying about 2 to 5 million years ago. And I don't think I have not yet timed the eye loss events. I don't think we know exactly when it started. I know some of the cavefishes themselves started diversifying 1 million years ago. Did it take the same amount of time? So in the previous investigation in Niemöller et al. 2013, the eye histology showed that the blind taxa lost their eyes in different ways. Although I'm not quite sure if that would take the same amount of time. Also, their rhodopsin data showed different areas of mutations and stop codons. So I'm not quite sure if they lost them at the same time or if it took the same amount of time. Yeah, that's a good question.
Yeah, that would be an interesting, interesting follow-up study for sure.
Definitely.
So we have another from, yeah, more work to do. Another from Winnie.
Do you know which genes are responsible for these morphological significant difference? I do not. I am hoping in my postdoc to get into some of the genomics of these fishes. And I think that that could be really interesting to look into exactly what the molecular basis is for why they have a duckbill snout. You know, I think that that's a great question, but no, I am not sure which genes are related to that.
And just as an aside there, that is something that's seen in some other cave fishes, right? Like I'm thinking of the Cynocycloculus.
Mm-hmm. And then oh my gosh, I just had it. Some of the cave catfishes start having a duckbill-like snout. Yeah, it's and people have noted it before, but I haven't been able to find geometric morphometric studies. So it really does seem like a convergent trait. And so looking into that genomic basis would be awesome.
Right. And any idea about the functional significance of it? Like what?
So that's a great question too. So it appears that it is neotenic. So it looks like, you know, smaller cavefishes also have the duckbill. And it's hypothesized that it could be that a larger mouth allows them to capture more prey. It's also possible that somehow that shape is more dynamic to allow for the sensory system, the neuromasts, to feel changes in the environment more sensitively than other shapes. But again, those are just hypotheses, and I think that would be excellent to look into. Do I have more time?
Yes. Take 1 or 2 more questions, and then you feel free to answer the other ones in the chat once we move to the next speaker. Yeah.
You mentioned the potential for introgression versus ILS to explain the case of discordance in your phylogenetic data. What sort of approaches are you considering to distinguish between the two? Oh, that's a great question. So obviously it's really hard to distinguish the difference between introgression and ILS. I am hoping that with further phylogenetic networks for PhyloNet, I know that can help look into it a little bit more. And that was where I was going to start. But I still have some work I need to do on figuring out which it could be. Why do you think the cave forms have a thinner body type? Oh, that's a great question too. What is the adaptive advantage? So that's a really good question, especially because when you think about Astyanax mexicanus, the guys behind me, the Mexican blind cave tetra, they hold on to fat a lot. And you would think that that would be the better way to survive in a cave, to have all that fat and keep it. But it's possible that maintaining that metabolically for the amblyopsids might not be enough. And perhaps they don't store fat, and that's the reason why they have thinner bodies. But again, that's a really great question. I'm not quite sure.
Awesome, thank you. And that's a perfect segue into our next talk. So more questions for Pam, just put them in the chat, and Pam, feel free to answer them, or we can come back to them at the end. And with that, I'll turn it over to Rachel Moran, coming to us from University of Minnesota. Go for it, Rachel.
Okay, can everyone see this okay?
Yep, looks good.
Okay. All right. So today I'm going to be talking about blind cavefish some more, as we've heard already, some other great talks about blind cavefish and how they provide insight into mechanisms of repeated evolution. So yeah, this is a nice segue from Pam's talk into my talk here. So I'm currently a postdoc in Suzanne McGaw's lab at the University of Minnesota, and I'll be focusing on some recent work in the lab here today. So it's a really exciting time for evolutionary biology right now, because we're starting to understand the genetic basis of adaptive traits in some of our classic textbook examples of evolution. For example, in peppered moths, we've seen that the causal variant underlying color morph evolution is a tandemly duplicated transposable element that affects the cell cycle gene. In Darwin's finches, a haplotype that is linked to pointy beaks has been passed among species through introgression. And in marine sticklebacks that have repeatedly invaded freshwater environments, pelvic armor reductions have evolved via a mutation in a region of the genome that's more likely to break during replication. So for all of these systems, the mechanism helps illuminate why the evolutionary change appears to be so rapid. And in many cases, we might not have predicted the type of causal mutation. So picking apart the genetic basis of traits also has taught us more about the evolutionary process. And a big question in evolutionary biology is whether certain genes are more likely than others to contribute to adaptive change. And if so, what, if any, commonalities might exist among those genes? So being able to predict which genes are most likely to be targeted by selection would be helpful in interpreting evolutionary processes and patterns. Systems where repeated evolution has occurred offer really powerful means to investigate this question, because traits displaying similar phenotypes in response to biotic and abiotic pressures are often under strong selection. One of the most iconic examples of repeated evolution is resistance to tetrodotoxin, or TTX, that's appeared in multiple lineages of snakes. This is probably best known from the study system of the common garter that preys on newts in Western United States. And across this, the snake phylogeny, which is represented here on the right, there's multiple occurrences of TTX resistance in unrelated taxa, which are each represented here in different colors. And in each instance, these snakes prey on TTX containing amphibians. And what's really striking is that across the phylogeny, TTX resistant snakes have relied on the same gene, NAV1.4. And not only that, the adaptive variation in snakes is clustered in D3 and D4 P loops. So what stands out is that identical or functionally equivalent replacements occur at most of the same residues in snakes, as in NAV1.4 of tetrodotoxic pufferfish as well. So the usage of the same gene to accomplish this phenotype is remarkable, because most vertebrates have between 15 to 25,000 genes that can be turned on or off in combinatorial ways to different degrees, allowing for a multitude of options for creating phenotypes. And another sort of related interesting observation is that mutational screens in the lab can create phenocopies using different genetic mutations than what we see in the wild. So this has been seen for Arabidopsis flowering time and Drosophila bristle number. So mutational screens have shown us that a given phenotypic change can occur through multiple different underlying mutations, but nature seems to often use similar, if not identical, paths. And so parallel evolution of mutations in the same genes in different lineages suggests that there's something about the commonly used genes that may be lending them to be drivers of phenotypic evolution. And these systems with repeated evolution help us understand if genes are used more often than chance and why. So for example, there could be limited genetic mechanisms for producing the same trait due to developmental or structural constraints, or regions implicated in repeated evolution may have fewer deleterious pleiotropic side effects relative to others. So in the McGaw lab, we use the Mexican tetra, Astyanax mexicanus, to study the causes and consequences of convergent evolution. And so this tetra occurs throughout streams of central Texas and northern Mexico, and in caves in the eastern side of the Sierra Madre Oriental, which is a large mountain range in eastern Mexico that's continuous with the Rockies. And there are currently over 32 cave populations documented. I'm showing you the 5 that we use most often in the lab here, and then also a surface fish up here. And so you'll see that the phenotypes can be variable, but they all share similarities such as a loss of eyes and a loss of pigment. And one really important aspect of the system is that cave and surface ecotypes are interfertile. So this allows for classic crossing schemes to be used to understand the genetic basis of some of these complex traits. And this provides us with an excellent system to examine whether evolution used the same genes and pathways to change these phenotypes from surface to cave forms, and whether there are common characteristics of the genes that led to phenotypic change. Oh, and another thing that I'll note that I'm going to be coming back to is that in the literature, it's been shown that there are at least two lineages of surface fish that migrated to northern Mexico and then invaded caves. So this appears to have happened at least twice, we've seen. Here is just a short little video. We can see some of these cave ecomorph Astyanax here. And so several traits you'll notice include the lack of schooling, the lack of aggression to tank mates, but they can still find their way around their tank just fine. And what's responsible for this is their increased neuromass cells and enhanced lateral line system. So the increased neuromass cells, including those around the orbit, are thought to aid in cavefish being able to detect vibrations in the water. And cavefish also have an enhanced ability to detect dissolved amino acids in the water compared to surface fish. And this is facilitated by an increase in the number of taste buds around their head. And their metabolism is also slower compared to surface fish, which Pam had just mentioned previously. And so they lose weight less quickly and they have more fat stores. Okay, so together all these cave adaptations, including the retinal degeneration and pigmentation reduction and enhanced sensory system, and loss in sleep and reduced metabolism are referred to as troglomorphic syndrome. And so in addition to all of these really interesting phenotypes and the interfertile ecotypes, there's also some genomic tools in this system. So in 2014, Suzanne and colleagues put out a paper on a short read genome assembly of the Pachone cavefish. And with collaborators, we've recently put together a manuscript on the surface fish genome, which is up on bioRxiv. And this was produced with long reads and optical mapping. And this new surface fish assembly is up on Ensemble and NCBI presently. So understanding repeated evolution requires an understanding of some of the basic parameters of the evolutionary process, including how long ago populations diverged, how many independent phenotypic origins have occurred, the extent of gene flow between populations, and the strength of selection needed to shape phenotypes. So to try to begin to answer some of these questions in the system, our lab recently conducted whole genome resequencing for 47 individuals across 5 populations. And this included 3 caves, Molino, Pachone, Tinaja, and then 2 surface populations, Rascon and Choi. This work was conducted in collaboration with the Yaniv Brandvain's group, also at University of Minnesota, and led by former postdoc Adam Herman. So what we're seeing here in this tree built using the whole genome sequencing data is that there are 2 independent lineages of surface fish that gave rise to cavefish. And so this supports previously published work in the system. So that's nice. And Tom Kono, another previous postdoc in the lab, did some demographic analyses here, and we saw some important new insights from this work. So the two surface lineages are much younger than previously estimated. So here, Tom estimated them at about 257,000 generations ago, and we can estimate there's maybe about 1 generation per year. I think that's fair to say. And conversely, previous estimates with mitochondrial DNA put this split at between 3.3 to 8 million years ago. So that's quite a difference. And additionally, the cave and surface splits within each lineage, we've estimated at between 160 to 190,000 generations ago, which is also much more recent compared to mitochondrial DNA-based estimates that put it at 1.8 to 4.5 million years ago. So these new insights have some important implications for what signatures of selection we can detect in the genome. Okay, so these were the populations that I was just talking about, just to have a visual again here. And I mentioned earlier that there are at least 32 caves that have been invaded by Mexican tetras. So we worked on expanding our analyses to now include whole genome sequences, whole genome sequence data from 8 surface and 18 cave locations. So this is what I've been working on during my postdoc here, is this big population genomic analyses where we've undertaken this big sequencing initiative to expand and build on our previous analyses and hopefully gain some broader insights into the evolutionary history of these populations. And this includes 233 resequenced genomes and 24 new assemblies. So here I'm showing a population tree, and what we're seeing is that it looks like there are at least 3 independent origins of cavefish adaptation in this cave ecotype here, which I have shown in these boxes. And if we look at a species tree shown here built in SVD quartets using over 1 million SNPs across the genome in all of these populations, we see that there might be potentially up to 5 independent origins of cave adaptation. So the take home here is that there could be between 3 to 5 unique evolutionary origins of cave adaptation. I'm currently working on digging into this a bit more and trying to resolve some of these relationships, which might also be heavily influenced by introgression, which I'm going to come back to in just a minute here. So understanding these phylogenetic relationships will play an important role in guiding our interpretation of other population genomic analyses. I'm currently investigating whether the same traits appear to be under selection in each cave lineage. For example, using HapFLK, which is a program that allows us to look, it's some software that allows us to look at haplotype frequencies across populations and detect regions under selection. And our whole genome sequencing data then allows us to dig into coding variants even after that. So we can ask whether the same causal variants are leading to cave adapted traits across independent cave lineages. So something we want to be aware of when we're looking for signatures of convergent evolution is that when we see the same mutation occurring repeatedly in the same gene, it raises this question of whether these mutations have evolved independently or have potentially evolved once and then spread via gene flow. So this has implications for how we understand the origin of phenotypic evolution and the repeatability of evolution in completely independent events. So an obvious question is how much gene flow is occurring from among Astyanax populations. And we know that caves are pretty harsh environments with no light and little food. And most surface fish that find their way into caves, like this one, end up in pretty poor condition. So there's this question of whether or not surface fish can survive long enough in caves to spawn. So going back to those 2 lineages that we focused on for the smaller population genomics study led by Adam Herman, analyses testing for historical and contemporary gene flow among populations found that gene flow is occurring within and between lineages, not just between cave and surface fish, but also among caves. And furthermore, while most caves contain low trait variability, presumably due to strong selection, there are at least 3 caves that have fish with really high levels of phenotypic variation. And so people have hypothesized that this may be due to newer invasions and evolution in progress. But what we're seeing is that it looks like these caves are actually more recent hybridization events, potentially hybrid swarms, and hybridization's been ongoing between surface and cave fish in these really diverse caves for over the past 1,000 to 2,000 years. So we're using these highly phenotypically variable hybrid populations to learn more about the genetic basis of traits maintained in caves. Okay. So just to summarize, cavefish provide a really powerful system for studying repeated evolution and can be used to investigate some big questions in evolutionary biology, including whether shared genetic architecture underlies the evolution of adaptive phenotypes, and whether adaptation is more likely to occur via selection on standing genetic variation, introgression, or new mutations. And so our population genomic analyses are still in progress, but so far have revealed that cave adaptation has evolved multiple times, and that gene flow is much higher than previously thought, both among caves and between cave and surface populations, which has implications for our understanding of repeated evolution. So I would just like to thank all of the members of the Astyanics community, including Suzanne and colleagues whose grants have funded this work, and all the members of the McGaw Lab and the University of Minnesota community. And I'm happy to take any questions.
Thank you so much, Rachel. That was fascinating. So we have a first question from Bridget.
Okay, yeah. So in the Mexican tetra, why do you hypothesize, what do you hypothesize has resulted in a large discrepancy in divergence time estimates for nuclear versus mitochondrial genomes? So I guess one thing, it could be introgression and gene flow between these lineages could be messing things up. Yeah, we're trying to dig into that a little bit more. So the first thing that comes to mind is potentially issues with gene flow. And also, you know, I mean, if you have whole genomes, you have more data. So, and their genomes versus mitochondria are recombining at different rates. So there's a few different things that could be at play there, causing those discrepancies. But yeah, it was a large time difference. Definitely shocking. Okay, next question from Ben. You mentioned that the hybridization in the swarm populations had probably been going for 1,000 to 2,000 years. How did you arrive at that time scale? So I used a hidden Markov model to get estimates for ancestry track lengths, and then we were able to extrapolate onto generation times, basically, and time since admixture using those track lengths. Yeah, so, okay, and then from Darko, how stable are these caves as suitable environments? Could it be that high extinction rates have shaped the phylogeny? This could be a confounding effect to study repeated evolution of the eye loss, and repeated colonization events. Yeah, that's a really good question. I know that in at least one of the hybrid caves that we study, it does seem like there have been some recent geographic events that have caused some changes to the environment. And so, yeah, that is something that we need to be aware of. But I think that some of the demographic analyses, like the ones that I showed by Tom Kono, a former postdoc, sort of help us rule out those sort of alternatives. Yeah, no, but that is definitely something to be aware of and consider, and to be aware of, yeah, the geographic history of all these areas too.
Kind of along those same lines, Rachel, when you've been doing the demographic analyses, have you looked at bottlenecking that might accompany colonization events? Is there any evidence for the cave populations that they undergo significant bottlenecks?
Yeah, so I think that we do have some evidence of that in some of the populations. That would be a great question for Tom. He did the demographic analyses. I think that was like right before I started as a postdoc. But yes, yeah, I think that, yeah, some of those populations definitely were invaded by smaller numbers of fish that were then able to adapt and maintain their numbers there.
Great. Okay.
Thanks.
Thank you. So we have our final talk. It's from Matthew Aardema from Montclair State University. So take it away, Matthew.
Sorry about that. You think after a whole semester and a half of lecturing, I would know what the heck I was doing, but here we are. All right, take two. Okay, so now hopefully you can both see and hear what I'm saying and showing. Okay, so we've been hearing a lot of talks about fish that have evolved in cave environments. And so I think kind of Liz put me last because I think all of you really have set up my talk quite well. So thank you for planning that so carefully, Liz. This is what I'm going to present is collaborative work with Melanie Stiasny and Liz Alter here, and this was recently published earlier this year in GBE. Okay, so as I said, we've really kind of been thinking about and talking about fish evolution in environments that look like this, very dark, somewhat stable, kind of, tranquil environments. So when we think about the Lower Congo River, it is probably perhaps the most kind of divergent habitat we might think of from the cave environment. It's notorious for having these incredible rapids, very dynamic hydrology, a really just kind of a very incredibly just distinctive and unique environment. And yet, as you probably can gather, from this environment, there is a cichlid species that has evolved that looks very much like a troglomorphic fish, right? It looks like a fish that if we didn't know where it came from, you would say, oh, that came from a cave, right? And when we compare it to kind of the canonical cave fish, of course, we see these similarities. We see, of course, the absence of vision-forming eyes. We see the apparent lack of melanization, so these very kind of white fish. And when you compare them to either surface populations in Astyonyx or in closely related similar or the same genus, fish of the same genus in the cichlids here in Lamprologus, those troglomorphic phenotypes really become quite clear and quite distinct. The lack of the eye again, and the lack of pigmentation. So when Melanie and Liz and I kind of set out to think about this fish and really think about how we wanted to go about assessing it, kind of one of the big questions we wanted to address is, do we see the same genetic changes in these fish that we would see in these cave-dwelling species? So Astyonyx, of course, being probably the most well-studied with all the genome resources and things available for it. There's a lot of kind of background knowledge about the underlying genetic underpinnings that result in this troglomorphic phenotype. And so to put this kind of question another way, do we see genetic convergence, right? And this is a question that has been brought up earlier today. And that's really the heart of what we want to think about. Do we see genetic convergence? And kind of a second component of that question really is to what extent and at what level do we see genetic convergence? And convergence can happen, of course, in many ways. So you may see the same variants, the same, SNP, the same indel in the exact same location, the same gene in certain instances. You may see convergence being driven by changes in the same gene, but through different mechanisms. You might have a SNP in one location in one species and a SNP in another location in another species, same gene, different SNPs, but they result in the same phenotype. And then kind of the furthest out and kind of where it gets to be a little bit nebulous, whether you could consider it genetic convergence, is looking at different genes, but which are believed to have the same kind of function, and really the protein that is encoded by those genes, whether those have the same function. And then do you see changes in those genes that may explain this convergence in phenotype? And broadly speaking, from other studies and lots of information, we have this idea that as we increase in the amount of evolutionary divergence we observe, we're going to get away from specific variants and really kind of go more in this direction. And so the cichlid here, the lithops, and then other cavefish, Astyonyx most specifically, are several 100 million years diverged. So we might predict we're going to see things more towards this end of the spectrum. So in terms of what we did, Liz has these samples. She extracted DNA from these samples. So we have our lithops at the top, and we also have this closely related congenitor, the Lamprologus tigropictolus, as kind of our outgroup comparison. We did the standard Illumina sequencing to about a 90x coverage in depth. And then we took that data and we mapped it to what was, and I believe still is, probably the closest available cichlid genome. So Neolamprologus burchardi, it was available, it was annotated. So we took our data from both of these Lamprologus species and mapped it to burchardi. And then we basically just called SNPs. To look at variation, SNPs and I should say SNPs and indels, so both types of variants. The next thing we did, kind of in parallel, was think about the genes that we would predict to see potential variation in lithops that could correlate with these troglometric traits. I already mentioned pigmentation loss and eye loss. We also looked at metabolism and feeding, circadian rhythms, and UV repair. Of course, if a fish is living in a completely lightless environment, it probably does not require a lot of UV damage repair. So we looked first at zebrafish, and we essentially found all the Go terms that were likely associated with these 5 phenotypes. And then using those, we then found all of the zebrafish proteins that correlated or were known to have effect on these traits. And then with that, we compared those proteins from zebrafish to those of the annotated Neolamprologus burchardi genome and found likely orthologs. So genes in Neolamprologus burchardi that likely influence these phenotypic traits. Okay, then, as if that wasn't enough, we took our matrix of variants that we observed in lithops and we observed in tigropictolus, and we annotated all of those. And in particular, what we were looking for were variants that were likely inactivating. So variants that probably, or we could postulate, likely broke the gene. You know, you often would hear these referred to as loss of function mutations. And the 4 specific ones that we focused on were the presence of a premature stop codon, the loss of a start codon, frame shift mutations. So these would be small indels typically that would result in kind of the protein shifting in the amino acid structure, and then changes to the exon intron splice sites, which again would likely break a gene. And I'm not going to show you a big long list of genes that we found, but we found a fair number of candidate genes, things that were present in lithops, but not in the normal fish, the tigropictolus. And we confirmed as many of these as we could through several mechanisms. We independently, with the data, did de novo assemblies to see if in the de novo assembly we recovered these inactivating mutations. We also did a lot of comparison to all the available cichlids and other species. That was really done to ensure that the annotation of Burchardi here was correct. In some cases, it had been annotated a little funky, and it would seem in relation to other cichlids. And then for a subset of the genetic variations and the inactivating mutations, we actually did PCR with additional lithops samples to confirm that those inactivating mutations were in fact present there. So we found a lot of stuff. I'm not going to go through all of it. I'll just kind of highlight two of the key findings. So the first key finding, perhaps arguably the most interesting finding, was the loss of the start codon in the OCA2 gene. So what I want you to see here is this, whoops. Again, you'd think I would know what I was doing by now. There we go. All right, so this SNP, this single nucleotide polymorphism right here, this is in our lithops, and in all our other species, we have a T. This is the start codon for the OCA2 gene. So this is 5' UTR here, and this is the very first exon of this gene. And so this simple single nucleotide mutation from a T to a C basically gets rid of the methionine, which is our start codon, and replaces it with a threonine. And so we don't have necessarily functional data, but this is really good evidence to suggest that the OCA2 gene is basically, broken in lithops. And, this is a gene that is widely observed to influence coloration in a wide variety of organisms, including Astyonics. So there's lots and lots of research on this gene in Astyonics. I've kind of semi-randomly picked a paper here. There were papers looking at this gene in Astyonics before this one, and of course subsequent papers as well. But I just grabbed this paper here just because I like this figure, just showing how in Astyonics it is the loss of an exon that correlates with the loss of pigmentation in the cave variants of, or the cave populations of Astyonics. So same gene, same phenotype, but seemingly a very different mechanism. Moving on to findings regarding the loss or reduction of the eye in lithops. So we found a number of genes, about 11 or so, that again had inactivating mutations, loss of function mutations. We don't necessarily want to go through this full list. What I do want to point out is the number of crystallins that we found. Crystallins are a protein that are really important, as the name might suggest, in the formation of the lens. And again, lots of work in Astyonics. This is just a review chapter in a book that I grabbed, again, mostly because I could pull this figure out of that chapter. And this is a really good kind of summary of the large number of genes that have been examined and appear to have some correlation with the loss of eyes in Astyonics. And of course, you wouldn't be able to tell this necessarily, but one of the really interesting things we found was that there was actually no specific genes that overlap. So of all this large list of genes in Astyonics, we didn't actually find any high confidence, the same gene with high confidence with an inactivating mutation. But go back to kind of how I started the talk and thinking about the nature of convergent evolution, given that these species are hundreds of millions of years diverged, it perhaps is not that surprising. We do see the broad similarities in the type of genes. In particular, the crystallins that we observed are also, there are several crystallins in Astyonics, for example, that also appear to influence the loss of the eye in that phenotype. Okay, so to kind of wrap up, what can we say? What did we see? What did we find? So with pigmentation, we actually see it's the same gene that does appear to influence the loss of pigmentation, the loss of melanization, in particular the OCA2 gene. So here we see it's the same gene that has converged to result in this troglomorphic-like phenotype in the cichlid. With eye loss, it really seems to be more in this realm where there are so many genes responsible for the formation of the eye and so many things that can start breaking when the eye is no longer needed, that we don't necessarily see a lot of overlap in the specific gene realm, but rather we just see these broad categories of genes starting to kind of become inoperational in these species. So with that, I will conclude, and I will simply just say for part of my time in this research, I was provided by a Gershner Fellowship in Bioinformatics and Computational Biology while I was at the American Museum, and I will take questions if there is time.
Great, thank you, Matthew. And I should mention too that Matthew his specialty is insect genomics, but he bravely waded into this study, and I think after that we can consider him an honorary ichthyologist.
Thank you.
All right, so we've got one talk to begin, or one question to begin. Sorry, from Darko.
Okay, related to the genome assembly method, what if mapping against the same reference for both species is masking genomic rearrangements on lithops, rearrangements which could result on truncations of genes, for example? Do you find any of this when running the de novo assemblies? So there's a couple of kind of challenges with the specifics of this. The biggest one is that the Burchardi genome assembly is of questionable quality. So right away that made things pretty challenging and limited kind of the confidence we had in what we were finding. And then with the de novo assemblies, I guess there's kind of two ways to answer your question. One, I would say we didn't necessarily look extensively for rearrangements. You know, given that we only had the short data, it would have been challenging even if we did find something to really put a lot of weight on that and consider it to be, you know, high confidence. Kind of the follow-up to this study, and I kind of excise those slides because of time constraints, is really to kind of get more samples and really just improve our ability to actually look for structural variation and rearrangements and things that surely are contributing factors. We are pretty confident that we are missing a lot of the kind of derived changes in lithops that are contributing to that troglomorphic-like phenotype. This was really that first pass where we could hopefully find those variants that do seem to correlate with the phenotype. So yeah, hopefully I answered the question. Any?
Yeah, let's see if any additional questions come in. And otherwise, we're about at the point where we can kind of wrap up, and I can turn it over. Well, first, let me once again thank all of our speakers. Amazing set of talks, and I think really highlighting how cool these systems are for thinking about everything from trait reversal to diversification and gene flow and demography, comparative genomics, all in these systems that have a lot of interesting similarities and some interesting differences as well. So thank you once again, everybody. And I will turn it over to Athena